This is an example of visible damage caused by branch rot in chili plants, often associated with stem twig rot caused by fungi such as Phytophthora capsicum or other fungi. In short, branch rot causes chili plants to wilt, rot, and often die, resulting in significant losses for farmers. This disease is especially detrimental during the rainy season and high humidity conditions due to its rapid spread. Now, let's discuss this disease more comprehensively. We need to understand how the disease originates, how it develops, and how it can damage chili plants, causing crop failure. We also need to understand the factors that cause its arrival and the specific reasons why the fungus that causes the disease can develop so quickly. Once we understand these two things, we must also understand the steps to prevent it from occurring, or be able to implement control measures appropriately, accurately, and economically. The disease commonly known to farmers as twig rot in chili plants is often a manifestation of a broader disease, namely stem, root, and fruit rot caused by Phytophthora capsicum leonian. Phytophthora capsicum is one of the most destructive pathogens in the Solanaceae and Cucurbitaceae families, causing significant yield losses, even up to 90% in cases of severe infestation. This pathogen is not a true fungus, but belongs to the Oomycetes group, which is phylogenetically closer to algae. This characteristic is very important in determining chemical control strategies. Phytophthora capsicum attacks all growth phases and all parts of the plant, holocyclic, including roots, base of the stem, stems, twigs, leaves, flowers, and fruit. Infection on twigs or branches often begins with rain splashes carrying inoculum, spores. Initial symptoms are blackish-brown lesions or spots that are water-soaked. These lesions develop rapidly and encircle the branches, girdling, cutting off vascular supply, xylem and phloem. When the flow of nutrients and water is cut off by the lesions on the branches, the plant above them, including leaves and fruit, wilts suddenly, even when the soil remains moist. The wilted branches then dry out, turn black, and die. If the infection spreads to the main stem, basal stem rot, the entire plant wilts and dies. On fruit, the infection causes widespread, water-soaked brown spots that cause the fruit to rot and drop. The life cycle of Phytophthora capsicum is highly influenced by environmental conditions, making it an epiphytotic disease, easily becoming an epidemic under certain conditions. The pathogen survives in the soil and diseased plant debris as oospores, thick-walled sexual spores, and/or chlamydospores, asexual dormant structures. When soil conditions are saturated with water and temperatures are favorable, the oospores germinate and form sporangia. The sporangia release zoospores, motile spores with two flagella. These zoospores can swim in a thin layer of water on the soil surface or irrigation water, which is the main mechanism of dispersal from the soil to the roots, base of the stem, and lower branches, via water splashing. In chili cultivation practices, several practices can also influence the development of this plant-damaging disease, including 1. Planting density. Planting too close together impedes air circulation, increasing microhumidity around the stems and branches, thus accelerating pathogen development. 2. Excessive nitrogen fertilization. Applying high doses of nitrogen N fertilizer can cause overly succulent plant growth, watery and soft, making stem and branch tissue more susceptible to pathogen penetration. 3. Former infected land. Continuous chili cultivation, monoculture, without crop rotation on infected land will increase the amount of Phytophthora capsicum inoculum in the soil. 4. Mechanical injuries. Wounds caused during cultivation activities, pruning, tying, or pest insect attacks, provide entry points for the pathogen to infect the branches and stems. To maximize the prevention and control of twig rot disease, it must be based on the principles of integrated pest and disease management, IPM. IPM combines various methods to suppress pathogen populations below the economic threshold without causing significant negative impacts on the environment. A. Preventive strategies. Prevention focuses on strengthening plant defenses and manipulating the environment to prevent pathogen development. 1. Cultural practices. Selection of resistant varieties. Scientifically, the use of chili genotypes that demonstrate resistance or tolerance to Phytophthora capsicum is the foundation of prevention. This pathogen has high genetic variability, so research is ongoing to develop superior varieties. Land and tool sanitation. Thoroughly clean up diseased plant debris, inoculum, and destroy it, for example, by burning. Furthermore, sterilizing agricultural tools, such as hoes and sickles, with disinfectant or soap before moving to another field is mandatory to prevent transmission. Crop rotation. Implement crop rotation with non-host crops from the Solanaceae family, e.g., rice, corn, or beans, for at least 2 to 3 years. This aims to break the life cycle of Phytophthora capsicum in the soil. 2. Environmental management. Optimal drainage. Phytophthora capsicum is exacerbated by waterlogged conditions because the pathogen spreads through waterborne zoospores. Therefore, creating high, dome-shaped beds, deep ditches, and ensuring excellent drainage are key to limiting the spread of zoospores. Plant spacing and humidity. Adjust plant spacing to avoid excessively dense planting. Ideal plant spacing will improve air circulation, aeration, between plants, thereby reducing relative humidity around the canopy and minimizing dew formation, which is a trigger for infection. Plastic mulch. Silver plastic mulch reduces rainwater splash from the soil onto stems and leaves, which is the primary mechanism for spreading inoculum from the soil. 3. Balanced plant nutrition. Avoid excess nitrogen. Excessive application of nitrogen N, a fertilizer, tends to produce succulent, watery, plant tissue, making plants more susceptible to rot disease. Use of ZA, ammonium sulfate, fertilizer. Several studies have shown that the use of ammonium sulfate, ZA, fertilizer can slightly lower soil pH to acidity, which can indirectly suppress the activity of Phytophthora capsicum. B. Control strategy, curative. Control measures are taken when disease symptoms are detected. 1. Mechanical control. Immediate removal and destruction. Plants showing symptoms of twig or stem rot should be immediately removed along with the roots and burned or buried away from the planting area. This is a critical curative measure to reduce the source of inoculum that could spread. Pruning infected twigs. If the infection is still localized on a twig or branch, cutting the diseased part down to healthy tissue can be done, followed by applying a fungicide to the cut wound. 2. Biological control, biocontrol. Antagonistic agents. The use of beneficial microorganisms, biological agents, such as the fungus Trichoderma and the bacteria Pseudomonas fluorescens or Bacillus subtilis, has proven effective. Application of these agents, usually during basal fertilization or seedling treatment, can occupy the ecological niche around the roots. Produce antibiotic compounds that inhibit pathogen growth. Promote induced systemic resistance, ISR, in plants. 3. Chemical control. Selective fungicide application. Fungicide use is a last resort and must be carried out judiciously to avoid pathogen resistance. Systemic fungicides, phenylamides, e.g., metalaxyl, are effective because they are absorbed and distributed throughout plant tissues, working from within to inhibit the growth of Oomycota. Contact protective fungicides, coppers, e.g., copper hydroxide, or dithiocarbamates, e.g., mancozeb, are applied as protective agents to plant surfaces to prevent pathogen spores from germinating. Active ingredient rotation, rotating the use of fungicides with different active ingredients based on their mode of action, is important to minimize the risk of selecting resistant pathogen strains. Disciplined and integrated application of IPM, prioritizing cultural and biological preventative measures, is the most sustainable and effective scientific approach to mitigating losses from chili twig rot. Behind the thick leaves, there is a red-hot promise: "O persistent farmer, do not be afraid in the midst of the weather. Even though the rotten branches come, testing your soul's patience, remember, the roots of your spirit are much stronger than pain. This chili is not just a fruit, it is a crown of struggle. Every drop of your sweat is a life-giving prayer. Your hands are a fortress, your prayers are holy water. Fight the withering that surrounds you, care for the eternal green. Restore fragile branches, clean wounds on the stem. Because at the end of the branch, the smile of the harvest awaits the light. Your spirit is an unquenchable ember, your results are endless blessings. Keep fighting, farmer, because the chilies in your field are your pride."